JEV, or Japanese encephalitis as it is more commonly known, is a virus which can cause severe and even life-threatening symptoms in patients who have contracted the disease. It is the most significant of the encephalitis diseases, killing over 15,000 people annually. JEV is a genus of the common tropical disease-causing pathogen flavivirus, the same viral agent that's responsible for the Zika virus and yellow fever. It is spread by mosquitoes who carry the virus from infected animals to humans who are dead-end hosts. It is the most prominent disease throughout Asia and Northwest Oceania and is considered to be a rare exotic disease to the Western world. Japanese encephalitis kills 1/3 of its patients and leaves half of the survivors with permanent neuropsychiatric sequelae. But what makes this disease so effective in taking over its host's body? Or why does it have such a high fatality and impairment rate? Let's take a closer look. The human body has 3 lines of defense that work to keep foreign objects out of its systems. Because JEV is injected into the host via mosquitoes, it can evade the innate immune response, making the 3rd line of defense the most significant in fighting off the infection. The 3rd line of defense is composed of the cell-mediated and humoral responses that are responsible for stopping the pathogens from infecting the important internal systems. The cell-mediated response sends the macrophages to eat the infected cell, then to wear its remains as a symbol of what the invading cell looks like. This is known as the antigen. The T helper cells then attach onto the antigen on the receptor of the macrophage, where it turns it into cytokines. The cytokines are then given to the T memory cell, which also attaches onto the antigen on the outside of the macrophage. Once the memory cell has enough information of what the antigen looks like, it rapidly multiplies via mitosis into T killer cells, which then destroy infected cells. The humoral response is where AB cells become activated after they have had contact with a matching antigen. The B cell then engulfs the antigen and displays it on its outside. The antigen-covered B cell then attracts the attention of a matching T cell, which then secretes cytokines and helps the B cell to turn into a mature plasma cell capable of producing antibodies. Once the JEV virus has been injected into the host via mosquito, it is moved via passive transport to the lymph nodes. In response to an impending JEV infection, the body reacts with the inflammatory response, making the lymph node glands swell throughout the body. The humoral response is to produce antibodies and release them into the blood and cerebral spine fluid. Vulnerable populations, such as children, are less likely to produce antibodies quickly enough to control the disease. In some people, the JEV virus constantly mutates its antigens, especially its epitopes, to become incompatible with the antibodies produced against it. In addition, JEV is thought to restrict the release of macrophages or B and T lymphocytes as well as cytokines. It is thought that this is the reason why different people's immune responses differ. The virus then makes its way to the nerve cells, where it incubates for 5 to 10 days before emerging as the mature virus. The JEV virus is then transported via bloodstream to the brain, where it crosses and damages the blood-brain barrier and into the cerebroendothelium. Due to the blood-brain barrier being impaired, inflammatory cells leak into the brain, exacerbating the damage. Microglial cells, which are specialized macrophage immune cells in the brain, release neuroprotective factors to facilitate the recovery of impaired neurons in the response to the inflammation. The microglial cells also phagocytose dying or damaged neurons before they can secrete toxic chemicals. The death of these cells then triggers the microglial cells to release more neuroprotective factors, leading to a recurring cycle of inflammation and nerve death. JEV's ability to evade the human immune system, combined with the way the virus is spread, explains why JEV has reached epidemic proportions. Human vaccinations are considered to be the most effective way to combat JEV. However, as the human is a dead-end host, it is difficult to eliminate the disease this way. Formalin-inactivated versions of the virus developed from infected mouse brain tissue are injected into the person's body, which allow their immune system the time it needs to recognize the virus as a foreign cell. The cell-mediated response then takes over and begins to easily destroy the weakened virus cells by creating an antibody that works against the antigen. When the person next comes into contact with the disease, instead of repeating the same process, the T memory cells will automatically recognize the disease and begin producing the antibody needed to destroy it. The formalin inactivated vaccine, known as the Korean Green Cross vaccine, is used throughout Japan and Korea and is the only recommended vaccine by the World Health Organization. However, it does have some limitations. The vaccine requires multiple doses in order to maintain immunity and efficiency and is expensive to produce. This is a problem for people in developing countries with large populations such as India, as immunity is expensive and difficult to maintain. The conceptual model that I've chosen is effective at showing how vaccines reduce the impact of disease through the diagram of the immune system at work creating antibodies from the inactive version of the disease. The model also shows how vaccination of humans will not be effective at stopping the spread of disease, as the virus is spread by other vectors such as animals and mosquitoes and does not spread between humans.